Hi guys, Miss Francis here today to talk to you about genetic mapping. So using information from Morgan's experiment, his student Alfred Sturtevant used the crossing over of linked genes to develop what he called a genetic map. So what is this genetic map? It's an ordered list of the genetic loci or locations along a particular chromosome, where the frequency of recombinant offspring is reflected in the distances between genes on a chromosome. Now, the greater the distance between two linked genes, the higher the probability, the more likely that crossing over is likely to occur. Why is this? Because if I have a greater distance, then there are more points between the genes where crossing over can occur. So how is the map created using data from test crosses that were done on fruit flies? Well, you look at the recombination frequency, where that frequency is equivalent to this arbitrary number called a map unit. So for example, when they were looking at eye color and wing shape, there was a 17% recombination frequency between those two genes. That means that these two genes are located 17 map units away from each other within the same chromosome. I highly recommend watching Mr. Anderson's video about genetic recombination and gene mapping. He goes through Morgan's experiment again and explains how the genetic map is created using that information. So here, Sturtevant expressed the distance between genes as map units, where one map unit is equivalent to a 1% recombination frequency. And Sturtevant and his colleagues actually were able to map the entire genetic sequence of the Drosophila using that information. So these map units indicate relative distance in order, not precise locations of the genes, but more recent techniques show the absolute distances between gene loci and DNA nucleotides. At this point, I would encourage you to take a look at the Mapping Chromosomes practice problems in our Genetic Problems Mendelian Genetics packet.